Hello everyone, welcome back to Gray's Gyan. Today we are covering a fascinating topic that is directly related to sperm development. The topic is intramanchette transport and intraflagellar transport, or simply IMT and IFT. Spermiogenesis is the final phase of sperm development. During this phase, a round spermatid, a simple, round cell, undergoes a dramatic transformation to become a motile, fertilizing sperm. This transformation involves three major events. First, nucleus remodeling. The chromatin inside the nucleus condenses, and the nucleus elongates and reshapes into the characteristic sperm head form. Second, acrosome formation. The acrosome is an enzyme-filled cap that forms over the nucleus. It plays a critical role during fertilization, helping the sperm penetrate zona pellucida of the egg. Third, sperm tail assembly. The flagellum is built, including the axoneme, outer dense fibers, and the fibrous sheath. Now think about the logistics here. All of these structural proteins need to be transported to very specific locations inside the cell, at precisely the right time. So who handles this incredibly precise delivery? That is the job of our two key players, IMT and IFT. Before we can understand IMT, we need to understand a structure called the manchette. The word manchette comes from French. It literally means sleeve. And that is exactly what this structure looks like, a sleeve, or a skirt, that wraps around the elongating nucleus of the spermatid. Structurally, the manchette is made of microtubules and filamentous actin, or F-actin. It surrounds the nucleus from below the acrosome and extends downward. The most important characteristic of the manchette is that it is transient, meaning it is temporary. It assembles, does its job, and then completely disassembles. In mice, the manchette first appears at step 8 of spermatid development, and is gone by steps 13 to 14. Now, inside this manchette there are two types of molecular tracts. Microtubule tracts, where motor proteins like kinesin and dynein operate for long-distance transport. F-actin tracts, where myosin VA, MyRIP, and RAB27B operate for short-range, local transport. An important insight from Letty and Cyronen's 2016 review is that proteins can actually switch between these two systems, ensuring that cargo delivery is always timely and efficient. Now let's talk about IMT, intramanchette transport. In simple terms, IMT is the process by which cargo proteins are transported along the manchette. And just like the manchette itself, IMT is also temporary. It only operates while the manchette is present. So what exactly does IMT do? Three main things. First, it traffics proteins between the nucleus and the cytoplasm. This is called nucleocytoplasmic exchange. Second, and this is the most critical function, it delivers structural proteins to the head-tail coupling apparatus, or HTCA. Now, what is the HTCA? It is a complex junction between the sperm head and the sperm tail. Its job is to keep the head firmly attached to the tail, so that when the sperm is swimming, the head does not detach. If the HTCA does not form properly, the sperm head can literally separate from the tail during movement. This condition has actually been observed in mutant mouse models. And as you can imagine, this severely impacts male fertility. Third, IMT uses a dual system, both microtubule tracts and F-actin tracts simultaneously. In terms of motor proteins, kinesin moves cargo in the anterograde direction, from the nucleus toward the tail. Dynein moves cargo in the retrograde direction, from the tail back toward the nucleus. Myosin VA operates on the F-actin tracts for short-range transport. All of this was described in the landmark 2011 paper by Kierszenbaum and colleagues, when they localized GMAP210 and IFT88 proteins within the spermatid Golgi apparatus. Now let's move to the second pathway, IFT, intraflagellar transport. If IMT is about the manchette, then IFT is about building the flagellum, the sperm tail. Think about it this way. The flagellum is an incredibly complex structure. It contains the axoneme, outer dense fibers, and the fibrous sheath. All of these proteins need to be manufactured, transported, and assembled at exactly the right location inside the growing tail. IFT does this job by working like a trained system. IFT particles, which are large protein complexes, ride along the axonal microtubules, just like a train moves along rails. Anterograde movement, from the base to the tip of the flagellum, is powered by kinesin 2, specifically the KIF3A and KIF3B subunits. Retrograde movement, from the tip back to the base, is powered by cytoplasmic dynein 2. The IFT system consists of 22 proteins organized into two complexes. IFTA complex, 6 proteins, primarily involved in retrograde transport. IFTB complex, 16 proteins, primarily involved in anterograde transport. Let's highlight a few key IFT proteins you should know. IFT88, also known as Polaris, this is part of the IFTB complex. It is essential for sperm head morphogenesis and tail assembly. Importantly, IFT88 has also been detected inside the manchette, which directly links the IMT and IFT systems. IFT20, this is arguably the most fascinating IFT protein. It is the only IFT protein that has also been found in the Golgi apparatus. This makes it a bridge between the Golgi and the growing flagellum. GMAP210, this is a Golgi protein that co-localizes with IFT88 in the spermatid Golgi. This was first demonstrated by Kirzenbaum and colleagues in 2011. KIF3A, the kinesin motor subunit that is essential for both manchette function and IFT anterograde transport. Let's do a quick comparison of IMT and IFT side by side. Location: IMT operates inside the manchette, a transient structure. IFT operates along the axoneme of the developing flagellum. Duration: IMT is temporary. It only works while the manchette is present. IFT is active throughout the entire period of flagellum assembly. Key destination: IMT delivers to the HTCA and the nucleus cytoplasm interface. IFT delivers to the tip of the growing sperm tail. Cytoskeletal tract: IMT uses both microtubules and F-actin. IFT uses only the axonal microtubules. Anterograde motor: IMT uses kinesin and myosin. Virginia IFT uses kinesin 2, specifically KIF3A and KIF3B. Retrograde motor: IMT uses cytoplasmic dynein. IFT uses cytoplasmic dynein 2. And the most important takeaway from this comparison, both IMT and IFT share similar cytoskeletal tracts, motor proteins, and non-motor cargo adapters. This molecular similarity, called molecular homology, was first proposed by Kirzenbaum back in 2002, and has since been confirmed by multiple experimental studies. Now let's look at something that runs in parallel with the microtubule-based IMT, the F-actin cargo pathway. Inside the manchette, there are actually two parallel transport systems working simultaneously. On one side, the microtubule-based IMT pathway handles long-distance transport. The motors are kinesin and dynein. Key proteins transported include IFT88, GMAP210, and IFT20. On the other side, the F-actin-based pathway handles short-distance, local transport. The motors here are myosin VA, MyRIP, and RAB27B. Specific cargo adapters link the cargo proteins to the myosin motor. And here is the elegant part: proteins can switch between these two systems. This cooperation, similar to a relay race, ensures that every protein reaches its assembly site at exactly the right moment. This coexistence of two parallel systems inside the manchette makes it one of the most sophisticated intracellular transport platforms in the entire body. Let's now take a moment to recognize the researchers whose work forms the foundation of everything we have discussed today. The first name is Dr. Abraham L. Kirzenbaum. Dr. Kirzenbaum was at the Department of Cell Biology and Anatomical Sciences at the City University of New York Medical School. In 2002, he published what is considered the founding paper of this field. Intramanchette Transport, Managing the Making of the Spermatid Head, Centrosome, and Tail, in the journal Molecular Reproduction and Development. In this paper, he was the first to formally propose IMT as a distinct transport mechanism, and he noted the striking molecular homology between IMT and IFT. Then in 2011, Kirzenbaum, along with co-authors Rivkin, Trace, Yoder, Haycraft, Bornans, and Rios, published another landmark paper, GMAP210 and IFT88 are present in the spermatid Golgi apparatus and participate in the development of the acrosome-macroplaxome complex, head-tail coupling apparatus and tail. In the journal Developmental Dynamics, this paper provided the first direct evidence that IFT88 is present in the spermatid Golgi, demonstrated how disrupted IMT leads to tail defects, and supported the role of IFT proteins in HTCA formation. The second landmark contribution comes from Lehti and Saarinen, 2016. Mari Lehti and Anu Saarinen, from the Natural Resources Institute Finland and the University of Turku, published a comprehensive review in 2016 titled Formation and Function of the Manchette and Flagellum During Spermatogenesis, in the journal Reproduction. This review brought together all existing knowledge on manchette assembly and disassembly, IMT and IFT mechanisms, and their consequences in mutant mouse models. It remains one of the most cited reviews in this field. Now, how do we know all of this actually matters in a living system? The answer comes from mutant mouse models. Multiple knockout, or KO, mouse models have confirmed that when cargo transport is disrupted, male fertility is severely affected. Model 1, IFT88 knockout mouse. When the IFT88 gene was disrupted in male germ cells, the mice produced approximately 350 times fewer sperm than normal mice. The few sperm that did develop had extremely short or completely absent flagella. Sperm heads were also abnormally shaped. This directly confirms that IFT is absolutely essential for flagellum assembly and sperm head morphogenesis. Model 2, IFT20 knockout mouse. Disruption of IFT20 in male germ cells produced infertile males. Spermatid heads were abnormally shaped, axonemes were disrupted, and two important sperm tail proteins, ODF2 and SBG16L, failed to be properly incorporated into the sperm tail. This demonstrates that IFT20 is a critical link in the pipeline from the Golgi to the flagellum. Model 3, KIF3A knockout. Loss of the KIF3A motor resulted in a defective manchette, abnormal sperm head shape, and tail defects. This confirms that the anterograde kinesin motor KIF3A is critical for both manchette function and IFT-driven transport. The message from all three models is the same: disrupt the delivery, and you disrupt the sperm. Now let's put the entire story together in one linear flow, from the Golgi all the way to the mature sperm. Step 1. Golgi apparatus. Proacrosomal vesicles are produced here. IFT20 and GMAP210 are both present at this stage, helping organize and sort vesicles for delivery. Step 2. Manchette via IMT. These vesicles and cargo proteins travel along the manchette, using both microtubule tracts and effectin tracts, heading toward the nucleus and the HTCA. Step 3. Basal body and HTCA. Structural proteins dock at this junction. The sperm head-tail connection is established and stabilized. Step 4. Growing flagellum via IFT. IFT trains karyoxonomal and accessory proteins all the way to the tip of the growing tail. Piece by piece, the flagellum is assembled. Step 5. Mature sperm. The manchette is gone. IFT proteins are no longer present. What remains is a fully formed, motile sperm, ready for fertilization. And the clinical significance here is clear. A defect at any step of this pathway leads to disrupted cargo delivery, which leads to abnormal sperm morphology, which ultimately leads to male infertility. This has been directly confirmed by the knockout mouse models we just discussed. Let's now quickly recap the 6 most important points from today's video. Point 1. 2 pathways, 1 goal. IMT and IFT both exist to ensure that the right proteins reach the right place at the right time during spermiogenesis. Point 2. Manchette is a temporary highway. IMT only functions while the manchette is present. Once the manchette disassembles, IMT stops. Point 3. HTCA is the head-tail stabilizer. IMT delivers structural proteins to the HTCA. Without a properly formed HTCA, the sperm head can detach during motility. Point 4. IFT builds the flagellum. IFT trains assemble the sperm tail protein by protein. Notably, IFT proteins are completely absent in mature sperm. They are only needed during development. Point 5. Dual cytoskeletal system. IMT uses both microtubules and F-actin in a coordinated, switchable network. This gives the system remarkable flexibility and precision. Point 6. Clinical importance. Knockout mouse data directly confirms that disrupted cargo transport leads to male infertility. And that brings us to the end of today's video. IMT and IFT, the two delivery systems that build a sperm. If you found this video helpful, please give it a like, share it with your classmates, and make sure you are subscribed to the channel.